Hi everyone, welcome to this video. I am B. Clara P.T. Rodrigo, Assistant Professor, Department of Microbiology, Kamraj College. So in this video we are going to see about fungi in their systemic mycosis, and this is part 1. So first we see about the general introduction of fungi. Fungi, which is a unique organism, possess a diverse group of organisms in it, and they are non-motile, immovable, eukaryotic organism. If we take that cell structure, means it possesses a defined nucleus as well as nuclear membrane, and they may exist as saprophytes, which means living on dead or decaying matter, or parasites, which causing various parasitic disease, and depend on another living organism which was termed as host for their food material or life running. And then commensals, so which live normally inside the host without causing any harmful and sharing the mutual benefits. They lack chlorophyll pigments, but the term fungi came means in our minds we suddenly imagine the mushrooms, right? So most of the beautiful mushrooms possess attractive color, but this was due to some other pigments produced by that mushrooms, right? But they lack chlorophyll. Okay, so due to the chlorophyll lacking, they doesn't synthesize their own food, so they belong to heterotrophic organisms. Okay, and then they reproduce either by means of budding or spore formation. So it was otherwise termed as conidial formation or sporulations. So the spores which was produced by means of sexual or asexual mode. The fungal cell wall is made up of a rich polysaccharides termed as chitin. The first fungi were identified by Agostino Bassi in 1835 from silkworm disease called musaridin. The science which deals with the study of fungi is called as mycology. And then we are going to see about the major classification of fungi. The fungi is mainly classified into two distinct groups. So one is microfungi and another one is macrofungi. The microfungi are invisible fungi, or it may be seen only by using some microscopic instruments, but macrofungi we can easily see with our naked eye. So in that microfungi it was further classified into two sectors. One is yeast and another one is mold. So these yeast are, which was otherwise termed as unicellular fungi, and they are round or oval in shape, and their mode of reproduction is mainly by means of budding. In this picture we see many big as well as smaller cells. Here the small bud like substance was nothing but it's a younger one of that yeast cells, right? So this was a little protuberant from the parent cell, and later on it get enlarges, and after when it reaches certain shape or certain size, it detached from its mother cell and it forms a new yeast cells, okay, or progeny yeast cells. And then this is mold, and here the example for mold is Rhizopus. The mold is otherwise termed as multicellular fungi, which possess multiples of cells, okay, or many cells. Here these multicellular cells, they arranged as a in form of certain steps or fragments. They are termed as hypae. These hypae may be single or linear or branched. So they was otherwise called as, these hypae was otherwise termed as filaments, and the group of filaments or this group of fungi was termed as mycelium. And here this is an example for mold, and it was commonly called bread mold. Okay, its name is Rhizopus. So we can easily or clearly see on the surface of the spoiled bread. Okay, and here this is another example for mold, and this is mostly here one, nothing but Penicillium notatum. So the antibiotic penicillin was derived from this mole. Okay, and here is an example for macroscopic fungi, and this is nothing but mushroom, and this mushroom name is called Amantida, right? And now we are going to see about the morphological types of fungi. And based upon their morphological features, the fungi may be divided into 4 main groups. So first one is yeast, and these yeast, they are oval to round unicellular fungi which mostly reproduce asexually by the process of budding. And then the process here, I am going to explain about the budding process, the process in which cell develop protuberance at one side which emerges, and finally it separates from the parent cell and develop into new individual younger one cell. Well, and now we are going to see about molds. So the true form of mycelium structure, okay, the molds was otherwise termed as filamentous or mycelial structure or fungi. Well, here these true mycelium grow as a branching filaments called hyphae. So in this picture we are seeing this mold structure, and here the branching like structure was termed as hyphae. The individual branching structure was termed as hyphae, otherwise termed as filaments. So there are many filaments which was joined together and intertwined or look like a tangled mass. The overall hyphae was called as mycelium. Okay, these hyphae produce both asexual as well as sexual spores. Well, and sometimes these hyphae which possess some septum behind each nucleus, and the septum possess hyphae or termed as septate hyphae, and some hyphae they doesn't possess any septums or cross walls behind every nucleus. That absence of cross walls or absence of septum hyphae was termed as cenocytic hyphae. Well, and these molds, they cause many spoilage in food materials, and then they cause many infections to humans. Okay, the most probably aspergillus as well as mucor, and these are examples of some molds. And now we are going to see about yeast-like fungi. So name itself we can guess. So it possesses both the characteristic nature of yeast as well as molds. So basically it's a yeast, but its structure appearance resembles like a molds. And well-known examples Candida, it was a yeast, but sometimes the cell enlarged and it looks like a fake or false mycelium. It was otherwise termed as pseudomycelium or pseudohyphae. Well, and now we are going to see about the fourth sector of morphological forms, that is dimorphic fungi. So in the name itself we can here di means two as well as morphic mean morphological forms. Here these fungi exist either as a yeast or as a mold depending upon the growth conditions. Right? The yeast form, also called as parasitic phase, occurs in the host tissues and in vitro in the culture at 37 degrees Celsius. So in the temperature 37 degrees Celsius they exhibit in the yeast form. Okay, and while the filamentous forms, also called as saprophytic phase, occurs in soil at 22 to 25 degrees Celsius. So when comes to the temperature 22 or 25 degrees Celsius, they exhibit in a mold form. Most of the fungi causing systemic infection means the infection caused throughout the body. Okay, and belonging to this dimorphic category, examples is Histoplasma capsulatum, Sporothrix, Blastomycetes dermatis, and Coccoidum mycosis emanatus. And mycosis. So now we are going to see about the term mycosis. Although there are vast diversity in the fungi kingdom, only 100 species are of medical importance, and various human infection that causes usually termed as mycosis. So mycosis is nothing but fungal infection caused in humans. Well, and based upon the site of infection caused, these mycotic fungi was again classified into 3 types. So first one is superficial fungi, second one is subcutaneous, and third one is deep or systemic mycosis. Now we are going to see about the superficial mycosis. These infections occur in body surface. In other words, these are surface infection. that is infection caused on mucosa, skin, hair, nails. They usually don't involve underlying or deep tissue infection. Example for this is dermatophytosis, and in the dermatophytosis the examples are dandruff. Okay, common dandruff is caused by these superficial mycosis, and barber's itch. Okay, as well as itching or prurite sensation which was present in beard area, and then nail infection. It was otherwise termed as ringworm infection. It also caused by means of this superficial fungi. The second mycosis, we are going to see about the subcutaneous mycosis. Such infections are common and occur on the surface as well as subcutaneous tissues, right? And these subcutaneous tissue infection may lead to massive tissue distraction. The lesions may spread progressively through lymphatic system and also to other sites of the body. And an example for this subcutaneous mycosis is mycetoma. It was an infection caused in the subcutaneous layers or subcutaneous layer on the feet or on the leg areas. Okay, and then third one is systemic or deep mycosis. This infection is caused by the inhalation of fungal spores and from the lungs. Okay, so first after the inhalation of spores, the spore give birth to small vegetative forms of fungi, and these fungi which grow inside the lung, they colonize, they grow, they multiply, they colonize, and from the lungs they through the bloodstream they spread throughout the body, and this condition is called as disseminated. And here major of the systemic mycosis causing fungi are this dimorphic fungi, that is Histoplasma capsulatum, as well as Blastomycetes, Coccoidomycetes. Well, and this causes very severe damage as well as a fatal disease to particular patients. Mainly immunocompromised patients such as HIV patients or malignant patients or immunotransplant patients was affected by means of this dimorphic fungi most randomly compared to other age group, right? Thank you for your listening, and continuation of this video is in systemic mycosis fungi part.